thank you to BRIC and also to Cheon Laboratories for inviting Eurofins to participate in this webinar. I'm going to talk to you today a little bit about the growing healthcare and drug development challenge that we see in fibrosis. As mentioned, I'm the strategic content manager, and I just thought I'd take 1 minute to explain what that means, because it might be a role that some are not familiar with. So I work sort of at the interface and the nexus of the business development and the scientific leads and marketing and product management teams to make sure that we're presenting and delivering to clients everything that they are looking for. So I will go ahead and start. So what we'll cover today is really how fibrosis is a disease of inflammation and tissue remodeling, the impact of COVID-19 on fibrosis and also on drug development, and how we can drive actually success in development of antifibrotics. In particular, how we take this approach with Eurofins. We have an innovative mindset, which allows us to develop assays as needed. We have a team with great expertise, and you're going to meet key members of that team today in the Q&A. I'll introduce them later. And then, of course, we have our assays and services, and a big portion of this webinar will be kind of discussing what we offer in these areas and also presenting some interesting case studies. So first of all, let's look at fibrosis and how it really is a disease of both inflammation and tissue remodeling. And what we can see here is that we have on the upper portion of this slide some distinct mechanisms that contribute to fibrosis, regarding renal fibrosis, liver fibrosis, kidney fibrosis. And then at the bottom, we have some shared mechanisms. And as we go across from the left to the right, we have a increasing incidence of the disease. So really the main points of this slide are that fibrosis is reparative. So it starts as a repairing tissue involved in disease, and it's really the disease itself is initiated by inflammation. And you can see here some disease-specific triggers that occur at different cellular sites of injury. Also, we have the fact that fibrosis is really progressive. So as we move here from recruitment of different immune cells, we have permeabilization and increased permeability of epithelial cells and endothelial cells, depending on the tissue context. And we have activation of different signaling pathways involved in fibrosis. And ultimately, with this adaptive and innate immune response, we get to the point of where there's an extracellular matrix remodeling that happens, and we have this activation loop, which causes tissue stiffness, hypoxia, and epigenetic modifications. So again, we have distinct mechanisms which occur all across this pathway, and then we have shared mechanisms. So it's really interesting because the shared mechanisms are then what allow us to develop therapeutics that possibly can target all types of fibrosis. Looking at then the tissue-specific context, we can see that it really influences disease and therapeutic outcomes. We have a key role of T cells in this process. So here we have this instigator again, and inflammation, which initiates disease, and recruitment of T cells. And primarily we have Th2 T cells, IL-4 and IL-13, derived from these Th2 T cells, activate and are very pro-fibrotic. And then we have from the activated CD8 T cells, IL-13, which is very involved in inflammation and the pro-fibrotic disease. Some of these other T cells are less involved, and even the follicular helper T cells are controversial in whether they are fibrotic. And the Th1 cells actually are those that could be fibrotic or not. I'm sorry, I'm having a little bit of a technical difficulty, so it's distracting me here from the slides. When we turn to liver fibrosis, we really see that it starts as a disease of metabolic overload. In steatosis, we have accumulation of fatty acids via lipolysis, resulting in lipotoxicity in the liver tissues and liver cells. We then move on and progress with this lipid accumulation, which leads to an inflammatory response. And again, we see this dual pathway of the inflammation and then fibrosis. So in the liver, what happens is we have hepatic stellate cells. So these are the cells that then develop into fibroblasts, and we have then an extensive extracellular matrix remodeling, which occurs when HSC cells are activated. So the challenge for drug discovery and development for fibrosis is that there are some common pathways and also some unique challenges. So in common pathways, we see that actually fibrosis is on the rise. Even though it's a low incident disease, in the instances where we have fibrosis, it's very high prevalence. It can affect multiple organs. It does result in tissue stiffening regardless of the organ. Ultimately, this causes a loss of organ function. And in some cases, investigators are looking more at the EMT transition and its role in fibrosis. But some of the unique challenges we see for drug development, again, are the tissue context. For example, renal fibrosis and pulmonary fibrosis have unique considerations. And then, of course, the liver fibrosis is largely the result of metabolic dysregulation. In the pancreas, we have a distinct set of cells, the acinar cells, and damage, tissue injury in these cells has been linked to development of fibrosis, particularly there. Fibrosis, cancer can be an instigator of fibrosis occasionally, and then, of course, long-term inflammatory conditions might have unique signaling pathways leading to fibrosis. So we'll take a little bit of a tour of what the impact of COVID-19 has been on fibrosis. We can see that the pandemic caused by SARS is expected to significantly alter the global health burden. This is information and data that comes from the Institute for Health Metrics and Evaluation and the World Health Organization. Even before the pandemic, lower respiratory tract infections were the fourth leading cause of death worldwide. Of course, this is expected to rise. In addition to ARDS, or acute respiratory distress syndrome, we also have cardiovascular and long-term immune system and neurological even consequences. And so we have a critical need to develop additional treatments related to COVID-19. And so we'll look a little bit about what's happening in the lung. So what are the consequences of COVID-19 disease in the lung, and how does that relate to fibrosis? So investigators have seen some similar responses as to what happened in the 2003 and 2012 outbreaks of coronaviruses. And why is this so, especially with COVID-19? It's because the lungs are really the primary site of infection and injury then. It has a similar progression as renal, liver, and lung fibrosis. So there is, on the top here, we see the normal tissue, the endothelial cells lining the capillary, and the fibroblasts. And what happens then in COVID-19, there's an acute response to the airway epithelium. We have recruitment of immune cells, activation of the fibroblasts, proliferation, and again, remodeling of the extracellular matrix. So some of the similar mechanisms are at play. In addition, we can see that there are some unique characteristics for in terms of the onset. So we have irreversible scarring and remodeling of the lung that may occur. This occurs progressively, and it is common to several lung disorders, and including interstitial lung disease. And some of these are listed here. So pulmonary fibrosis as a disease can be very similar to COVID-19. We're going to touch on some assays that address some of these areas here, as well as in this previous slide. So how can we help in this challenging disease area from Eurofins? So we can do this by innovating. And here I'm just showing you one response we developed in-house to look at inflammation, and in particular fibroblast responses and adaptive immunity. This last area on the bottom is where I'd like you to focus. These are systems that we developed in response to client needs, and they demonstrate the adaptive immune system in regards to the effect on fibroblasts. So the T-cell effector responses in the context of peripheral tissue, it's related to fibroblastic disease. And then we have readouts in the system where clients can look specifically at biomarkers that are associated more with COVID infection and lung fibrosis. So this is just one example of innovating for the time. We also have a broad portfolio, and some of what's listed here allows us to look again at this dual disease. So we can target inflammation or extracellular matrix remodeling, or both, with phenotypic and cell-based approaches. Some of the signaling molecules that we investigate are caspase-3, PDE4, the toll-like receptors, and cytokines and chemokines. I won't have time to show you any case studies for all of these, but they are available. And then, of course, we will look at some of the in vivo models that we have for lung fibrosis and also liver fibrosis. Another strength of Eurofins as a CRO is our team of subject matter experts. And this is where I'd just like to introduce those teammates who are available for the Q&A. So we have Jennifer Drake, who works with the phenotypic team, and she's a study director based in St. Charles. She actually lives in Seattle, Washington. I work closely with her through the Phenotypic Center of Excellence. We have Jimmy Bao, who is the Global Technical Director for Pharmacology Discovery Services, which is a partner lab of Eurofins, working on development of the in vivo models. We have Phil Lin, a scientist in Taipei, who's been involved as well and available for Q&A. and Pupurchi as well. So let's look a little bit at our assays and services. So we'll start with liver fibrosis. Again, we see this, the causes are the stellate cell activation, collagen deposition, and inflammation that occurs as a result of activation of these chemokine receptors recruiting immune cells into the site of injury. CCR2 and CCR5 are important. They've been shown to, if the inhibition of these receptors can suppress the stellate cell activation. They're very involved in the macrophage response, which is key in fibrosis. GAL3 has been shown to also, where inhibition of GAL3 can suppress hepatic stellate cell activation. And then, in addition to looking at stellate cell activation in fibrosis, we can also assess extracellular matrix remodeling. LOX-L2 is a key enzyme that promotes ECM fiber remodeling here, and inhibition can reduce remodeling and reduce the impacts of fibrosis. So these dual impacts can require dual therapeutic strategies. So again, focusing on the progression of NASH, we can see that targeting hepatitis and fibrosis, where the inflammation and the fibrotic response are more pronounced, we can see we have therapeutic strategies that address these areas, and we're going to focus on, show you some data on CCR2, CCR5, and BAP1, and also for ECM remodeling. These additional therapeutic targets are available. All these targets that have been developed, all these assays that have been developed, used drug candidates that are in clinical trials for validation. And you can see, especially those two that are highlighted are the two we're going to focus on. I'm sorry, I continue to have some technical issues here, so I apologize. So some of the target-based finding and functional assays available are the CCR2, again, the chemokine receptor binding, the VAP1 inhibition, and the LOX-L2 inhibition. And you can see here with these assays, we can distinguish increased potency of ligands of the treatment by looking at the IC50. So MCP1, the impact on MCP1 is by far greater than on MCP3 and 4. Using these compounds, PX42472A, a VAP1 inhibitor, is currently in phase 2 trials for NASH. This other candidate is the LOX2 inhibitor. It's in phase 1 trial for lung fibrosis, and these are available. And if you have questions on these assays, we can ask in the panel afterwards. In addition to target-based assays, we have phenotypic assays. The benefit of phenotypic assays is that the readouts are closer to clinical outcomes. You can see here that target-based approaches enable rapid discovery of compounds on specific targets, as I just showed, but also the phenotypic approaches enable kind of an unbiased discovery of molecules that are active on these biological outcomes. And this is very key for a disease such as fibrosis that has a complicated phenotypic outcome and the dual roles of inflammation and extracellular matrix remodeling. Here is one example of a phenotypic assay where we can see that the model is TGF-beta induced collagen 1 expression in these human hepatic stellate cells. And you can see on the top panel here, these are the cells treated with vehicle. This is collagen induction with treatment of TGF-beta. The red is a staining for the collagen matrix deposited in the cell monolayer. And then you can see treating the cells with these inhibitors that are available, we can see a reduction in the collagen. So this is a single readout assay, and the power of this assay is that it actually is looking at endogenous collagen 1. And you can see here this can be quantified through a high content assessment. We also have, through the Phenotypic Services Center of Excellence, various assays. We have the Biomap Platform, Biomarker Services Platform, OncoPanel, and also quite a few custom services. I won't have time to talk too much about the biomarker services, but basically for any toll-like receptors or TGF-beta assays or assays for other cytokines or chemokines, we can do custom or off-the-shelf programs and projects for clients. So I'll talk a little bit about the Biomap platform and how this can be used in the development of antifibrotics. So the Biomap platform has 3 distinct pillars. The first is these human primary cells and co-cultures. They model tissue and disease biology. We also have predictive analytics. So clients can look at toxicity of compounds. They can do similarity searches with standards of care and also benchmarking and for competitive intelligence. We have in a database of profiles then, clinical stage, approved and failed drugs and chemicals, and also biologics. So this is not just for small molecule discovery. And the benefits for the clients and programs are that Biomap provides a high level of relevance. It can provide translational and predictive insights. And again, this database is constantly updated, so we call it a living database of key benchmarks and standards of care. So key to this, the Biomap platform are these disease models, which are made of human primary cell types. Many of them are in co-cultures, and they're stimulated to mimic a certain disease. And then we have selected readouts that are translationally relevant. So these are all protein readouts. The assays overall are very robust. The data are reproducible. And since we're benchmarking to clinical standards of care, they're very relevant for disease indications. I'll spend a little bit of time on this slide. Again, just to orient you, this is an example of a Biomap profile. So these are the systems that I just mentioned to you across the top. In each of these verticals is the data from the translational biomarkers of that system being treated for the disease, with the therapeutic against fibrosis. So this is a broad panel which reports on inflammation, immunomodulatory, and tissue remodeling biomarkers. And right here, and also cellular hemostasis. So 4 categories of biomarkers. We have some of the biomarkers indicated in orange, which can report, for example, on oncology, and then biomarkers, many of them indicated here in green, which can report on tissue remodeling. So even though this is not a specific project that was related to fibrosis, I'm showing it just as an orientation to what happens when these systems are treated with a candidate therapeutic. There's a report out of these different biomarkers. They're annotated, And we can get some information on multiple indications. So let's take a specific example of looking at fibrosis in a different setting than what we've described. So this is not renal or pulmonary or liver fibrosis, but something different, which is glaucoma. So in glaucoma, the trabecular meshwork tissue, well, in the normal state, it's under constant mechanical stress. And this constant remodeling of the tissue and of the cells and of the extracellular matrix is critical to maintain a normal intraocular pressure. In a disease state where there may be inflammation, excessive deposition of the extracellular matrix can sort of plug this output of the aqueous humor and cause hypertension. And then this, of course, can result in damage and ultimately blindness. So there is a group who actually did take the Biomap system, and they looked at their candidate therapeutic and its impact on intraocular pressure that was in an animal model, and then also looked at extracellular matrix molecules. So you can see here, to get an idea on inflammation and tissue remodeling with one type of service, this group used their candidate therapeutic on the Biomap Diversity Plus. And you can see they identified that their candidate therapeutic decreased some key inflammation-related activities, as well as increased some immunomodulatory activities and had increases on tissue remodeling. We've also had programs in interstitial lung disease and idiopathic pulmonary fibrosis using some of our phenotypic assays. In this particular case, they're looking at a compound that can modify TNF alpha. And in the presentation are the links if people are interested to learn more. Another thing that the phenotypic assays are useful for is to really reposition. A lot of this has been happening in terms of COVID, especially at the beginning stages of the pandemic. Companies were really looking for drugs that were already approved that could help with some of the symptoms, especially the respiratory systems. So something that drug repurposing can do is to allow people to have a side-by-side evaluation of drugs in a systematic manner on experimentally validated disease models, so for ranking, and also this can be used to evaluate drug combinations as well. So there's quite a bit on this slide, but I will walk through it. This was the use of the Biomap Diversity Plus, which is here on the right. This was a case where a client was looking to reposition for a new indication. So they had a small library of compounds from their original disease indication, and they were looking to repurpose and pivot for IPF. So they wanted to do this, but also look in a series of assessments that would maintain the fibrotic disease phenotypes. So what they did is they did some broad profiling of their candidate, and they compared it to nintedanib and pirfenidone. These are, again, the power of the target-based assays that I described much earlier for liver fibrosis, as well as what we have for renal and kidney fibrosis and general tissue fibrosis, that we, in all areas, use these clinical candidates to validate our assays. So then these clients were able to rank to identify the sort of sentinel activities that are associated with approved IPF drugs, and then take their compounds and rank them based on these identified sentinel activities, and then take their top compounds and move to a more specific disease panel, which is the Biomap Fibrosis panel, and then to really look for compounds that were effective in lung and kidney fibrosis. So together, this allowed them to prioritize for a new indication with a large unmet need. This is what the fibrosis panel looks like. And again, the biomarkers here, translational biomarkers, report on myofibroblast activation, matrix tissue remodeling and wound healing, and inflammation. And this panel in particular is relevant for pulmonary, renal, or general fibrosis. I mentioned the in vivo models earlier, so through our partner lab, Pharmacology Discovery Services, clients have access to many of these different models for chronic liver disease, for NASH, liver fibrosis, and lung fibrosis. Depending on the model, there are various endpoint readouts, clinical symptoms, weight to body ratio, liver weight to body weight ratio, and there's other listed here. And these are applicable for a diverse range of therapeutic modalities. And again, we have the experts in this area to join us on the panel for the Q&A for those that have more in-depth questions on the in vivo model. So in this very brief, and I'm sorry, technically challenged presentation, I hope you've seen that fibrosis is really a disease of tissue remodeling and inflammation. Multi-pronged approach is needed that can be both target-based and phenotypic approaches. The disease is on the rise, so of course developers are looking for additional effective, safe therapies. So just to highlight some of the progress that's been made, and particularly in pulmonary fibrosis. So again, the standards of care, pirfenidone and nintedanib, were approved in 2014 for IPF. The approval was expanded to interstitial lung disease for nintedanib in 2019. And so we have in the field the belief that the future of IPF treatment may be in combination therapies. And this thought is based on the current diversity that's out there in research and discovery programs. So what is that pipeline? So as of 2019, this pipeline contains 132 drugs, which are in all stages of development. Approximately 2/3 of the drugs are in the preclinical stage, and 14 of the drugs, all with novel mechanisms of action, are currently in late-stage development. And throughout the presentation, you can find the links to some of the references on the slide. So overall, really the goal of drug discovery, of course, is clinical adoption. It's not just about assays validated with clinical compounds. It's about getting those compounds to patients. So through the whole portfolio of services that we offer, clients can look at efficacy, safety, superiority, and then ultimately approval and adoption. Eurofins, of course, is a large CRO. I talked to you a little bit about the in vivo services. Hopefully we'll have more Q&A time for that. I talked a little bit about the translational phenotypic services and the in vitro assays that are available for target-based assays. I did not talk much about the chemistry or safety pharmacology, ADME talks, and of course, all the custom assays. And actually for fibrosis, the benefit is the ability to do a custom assay. I'm sorry. We have services across global sites, which involve model development, collaborative opportunities. Our scientific team can provide guidance with portfolio direction and proposal preparation. Many of our services, in particular Biomap and others, offer data delivery calls after clients receive their data. And there are also publication opportunities available. So thank you very much for listening. I apologize for the distracting technical issues on my end. And if you have additional questions or would like to reach out, please contact Makoto Imatachi.